Hello, everyone, and welcome to today's webinar, Verification of Autoantibody Serology Tests, Practical Considerations for the Clinical Laboratory. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BioRad. To learn more, visit them at bio-rad.com. We encourage you to participate today by submitting any questions you may have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd like to now welcome our speaker, Dr. Melissa Snyder, the co-director in the Antibody Immunology Laboratory at the Mayo Clinic. Melissa, welcome. You may now begin your presentation.
Hello, everyone. My name is Melissa Snyder, and I'm an associate professor of laboratory medicine and pathology at the Mayo Clinic in Rochester, Minnesota. I'm so glad you were able to join me today for this webinar on verification of autoantibody serology tests, practical considerations for the clinical laboratory. I only have one disclosure to make, and that is that I have been a member of the Strategic Advisory Committee for WRFN. The learning objectives for this presentation are one, to be able to recognize inherent heterogeneity of autoantibodies and the impact this has on clinical testing. We're going to be able to describe the regulatory parameters that are needed for verification of FDA cleared and laboratory developed tests. We're going to look at specific approaches on how you might verify these different parameters specifically for autoantibody assays. And lastly, we'll talk a little bit about the drawbacks of using method comparison to assess accuracy for autoantibody serology testing. So as a way of introduction for my presentation today, I really want to be clear what we're talking about. And the assays that we're going to be focusing on today are ones where we detect an antibody that has specific reactivity against an antigen. Now, in the context of autoimmune disease, we'd be talking about a self-antigen, but these antigen-specific antibodies may also be detected against microorganisms in the context of infectious disease or against environmental antigens in the context of allergic disease. Now, if we're going to be really focusing on autoimmune diseases today and what the role of serology testing is, and it's important to point out that this is applicable to both systemic and organ-specific diseases. And if we think about the role of serology testing in these various types of diseases, we see that we have one application, which is diagnostic. However, it is important to note that these diagnostic application autoantibodies generally are not considered diagnostic markers in and of themselves, so we don't use them in isolation, but interpret the results in the context of other pieces of information, both laboratory and clinical. There are a small subset of autoantibody tests which are useful for monitoring, although it really is limited to only specific diseases and specific autoantibody reactivities. I think it's also important that we take a look at the different methods that are used for determination or identification of autoantibody assays. The basis of autoantibody serology tests really are to use a capture antigen, which will allow you to separate the specific autoantibody that you're looking for from other immunoglobulins that are present within the patient's sample. You can classify these assays as being either immune complex based, which is based on visualization of a complex that forms between the antigen and the autoantibody. And I would consider this really a direct detection of an antibody-antigen interaction. This is compared to immunoassay methods, which are really based on detection of the autoantibody binding to an antigen where you have a secondary label detection antibody. And this I think of as more of an indirect method of detecting the antibody-antigen interaction because you're requiring the use of a secondary or detection antibody. Looking a little bit more in detail at immune complex-based methods, you have gel-phase methods, which where you have formation of the immune complex in a solid phase, such as single or double immunodiffusion. And then you have more of the solution-phase methods, where you have formation of the immune complex in solution, such as you find with immunoprecipitation assays, nephelometric or turbidimetric assays. And then if we look at the immunoassays, we have indirect immunofluorescence as being one subclassification. We have line or dot blot assays. We have solid phase immunoassays, which there are many variations of, such as enzyme immunoassays, chemiluminescent or fluoroenzyme immunoassays. And then we have the multiplex immunoassays as well, which is kind of a subclass of solid phase assays to in a certain way of thinking, but we separate them out here because they are multiplex as opposed to more the traditional solid phase immunoassays. The other thing I'd like to touch on before we start talking about verification requirements is the characteristics of autoantibodies. And one particular characteristic I think we really need to appreciate is the fact that autoantibodies, unlike most other protein analytes, are heterogeneous in and of themselves. And this is true in terms of their epitope specificity, of their isotype class, as well as the differences in affinity or avidity the antibody might have for its particular target antigen. The other thing we need to remember is that when we are looking or we're talking about detecting an autoantibody, we're really talking about assessing for the repertoire of autoantibodies that a patient might have. This is not a single autoantibody, but rather a collection of autoantibodies. And this repertoire of autoantibodies, or this collection, may differ from patient to patient, but it may even differ within a single patient over time. The key takeaway here, though, that we need to appreciate is that autoantibody heterogeneity does have a very significant impact on both the analytical performance of the method as well as its diagnostic characteristics. So we'll really be focusing primarily on immunoassays today. So I thought it would be relevant to just think a little bit about the components of an immunoassay. So we have an antigen, which really makes sure that this is what's capturing your autoantibody. We really want to make sure that this is representative of the native antigen. This could be purified or recombinant, but somehow it needs to represent whatever native antigen the autoantibody is recognizing. We need to have some source of calibrator or control material, and the source of this material can be very important. Whether it's sourced directly from patient samples or whether it's patient material that comes from some kind of commercial source. The calibration scheme is also something that is very relevant for the performance of a method. This could be a calibration scheme that is established for each assay, such as you have in the solid phase, like an enzyme immunoassay, or it could be lot specific from a manufacturer, such as you might find on some of the more automated platforms. And then we have the detection system. We can think of the detection system in two components. We have the isotype specificity, whether it's only detecting IgG, IgM, IgA, or perhaps a combination of various isotypes. And then we have the label that is found on the detection antibody. And I think this is primarily relevant for the analytical measuring range. And the methods of detection that we commonly see are colorimetric, luminescent, and fluorescent. The other thing I want to touch on, idea to touch on before we get into verification requirements, is this idea of quantitation. So if we look at assays that are qualitative, these are assays where we report a result as positive or negative. And this is this identification of positive or negative is usually in relation to some sort of cutoff value. Now we're going to differentiate that from quantitative methods, where you report a concentration in an absolute unit, such as milligram per liter, something along those lines. These quantitative methods are generally traceable to some sort of certified reference material or CRM, or possibly some reference method. In between qualitative and quantitative, we have our semi-quantitative assays. And how do semi-quantitative assays differ between qualitative versus quantitative? Well, with semi-quantitative assays, usually these are going to be reported in some kind of relative concentration, such as units per milliliter, or perhaps even a titer. In addition, semi-quantitative assays generally are not traceable to a certified reference material or to a reference method. And when we think about autoantibody testing, generally we're thinking about semi-quantitative assays. However, I would like us to kind of think more beyond just that we are measuring concentration, but really think that what we're really measuring is reactivity and not necessarily a concentration. And to keep in mind that the result that was reported does not necessarily equate to a true concentration, because there are so many other parameters that impact the reactivity that you might detect in an assay, such as the affinity of the antibody for that particular antigen, and not just solely related to how much antibody is present within a patient sample. So we need to think about regulatory classifications. And when we think about this for laboratory tests, many of you, I'm sure, are very familiar with this. We have FDA cleared, where the FDA has reviewed data for a test and approves that it be used as an in vitro diagnostic. And usually when laboratories are performing FDA cleared assays, they are using the manufacturer's instructions. Now, a laboratory may modify an FDA cleared assay. They've made some sort of change in how they perform the testing. They've deviated from the manufacturer's instructions, and these would be considered FDA modified tests. And then we have the laboratory developed test, where the clinical laboratory itself develops and validates the method for the purposes of testing patient samples. So what are the regulatory verification requirements and that we have to follow between these different types of assays? For FDA cleared, we have the 4 primary parameters of precision, reportable range, or analytical measuring range, or AMR, the reference range, and accuracy. For FDA modified, or LDTs, we still have to fulfill those 4 primary requirements, but in addition, there's analytical sensitivity, analytical specificity, and then CLIA also highlights that any other characteristics that are required for test performance may need to be assessed as part of the verification for an FDA modified or LDT. For the purposes of the presentation today, just in the time limits that we have, I'm only going to be focusing on the 4 requirements needed for FDA cleared, which is the precision, reportable range, reference range, and accuracy. So let's start off with precision. Precision is defined as the closeness of agreement between independent replicate measurements. And this may include assessment of intra-assay or within-run precision and inter-assay or between-run precision. And usually the data is expressed as a % CV. The experimental approach is usually pretty standard. Usually we see replicate measurements of about 20 is usually considered appropriate. And this is true for within run and between run. And usually we like to assess precision across the analytical measuring range of the assay. So here's some example data that I generated in my laboratory. This is for anti-Saccharomyces cerevisiae IgG antibodies. You can see that we've selected 3 samples, different reactivities, which are shown here. The mean reactivities shown here in this first row for the 2 samples. What we're showing here is intra-assay precision, and here's the inter-assay precision. You can see our standard deviation, and then calculating a % CV for each of those samples. Here's another example of some data from anticardiolipin IgM testing. The tables are set up in the same way. We looked at 3 samples, again, across the analytical measuring range, and then showing the % CV as our final calculation for both intra-assay and inter-assay precision. And I would say for autoantibody testing that the precision is one of the more straightforward verification requirements that we can assess for. However, I think there are still some important points to consider for precision when you're thinking about autoantibody assay testing. The first is the sample source that you use. We generally try to use patient samples or patient pools. I think this provides a more real-world evaluation, particularly because we're using that patient matrix. But in addition, it represents all aspects of the testing protocol, including the dilution of the patient sample that might be needed prior to performing the assay. Sometimes QC material can be used, and sometimes we do include it in our precision experiments, usually as an addition to our patient samples. And it can be useful as a comparison to give you a sense of how the QC material will perform in terms of precision as you begin to offer the testing clinically. Sometimes we will take QC or even calibrator material and spike it into a negative serum to make it more of a patient matrix. But again, it just depends on the availability of that material as well as the availability of your patient samples. The other thing that we do is say that usually we assess precision across the analytical measuring range. And I have kind of two thoughts about this particular kind of aspect of assessing for precision. One is I always ask this question of myself, but really, what is the value of assessing precision in a negative sample? Because really, I think what you're doing here is measuring the background. Does it give you a sense of what the precision is in that area? It does, but again, it's more background and not necessarily measuring precision of a low sample. And it may be more valuable to try to assess precision close to the cutoff of your method rather than trying to just use a negative sample. But I will say it is something that we do routinely. It's just something I always wonder whether it's something that we should be doing or whether it's something that is needed. The other issue is that we always also try to assess precision at the high end or the higher end of our analytical measuring range. But the plain fact is that sometimes samples with high reactivity may not be available, and it may be challenging to find samples where you could really assess across the AMR for precision. So next, that brings us now to reportable range. I'd like to cover this aspect of verification next. So reportable range is also known as the analytical measuring range, or AMR. This is the range of analyte concentrations over which specified criteria are met, such as precision and bias. Now, usually the AMR spans concentrations from the lower limit of quantitation to the upper limit of quantitation. And this is generally limited to the linear range of the assay, meaning that when you dilute a patient sample that you see a decrease in the concentration that you calculate with the appropriate dilution. So this can be done in two different ways for autoantibody testing, at least. We could do mixing studies where we take a high and a low sample when we combine them in varying ratios to measure, to assess for linearity. And we could do a dilution evaluation where you do serial dilutions of a high patient sample. What I'd like to do is show you some data from some actual verifications that we've performed in my laboratory, showing these different approaches and how they may differ, and how the different types of data that you might see. So first, I'll show you some reportable range data from tissue transglutaminase IgA antibody testing. And these were dilution studies that we performed using high patient samples. And we started with two different patient samples, one at a 35.7 results and one at an 84.2. And then we did twofold serial dilutions, which are shown kind of down the column here. We measured those results after performing the dilutions, and then we compared that to what we would expect for those samples based on the dilution that was performed. And we did that again for these two samples. And in order to keep the table, try to keep it a little cleaner, I'm not actually showing you the calculation of the percent recovery here, but what you can certainly see, I think, is that as the dilution increased, as we continue down with the serial dilutions, the difference between the measured and expected value on a percent basis becomes larger and larger. And that is true for both samples as we compare the measured result to the expected result. Here for sample 2, we can see these measured results compared to what we would have expected. They always tended to be higher as well. They tended to, again, increase in the percent difference between them as we increase with the dilutions. And so that's really what we saw whenever we did this dilution experiment, is that even though when we diluted the sample, we certainly saw a decrease in the measured reactivity that we were detecting. However, this recovery was not linear in comparison to what we would have expected those concentrations to be based on the starting reactivity of the sample. Now, we took a different approach with beta-2 glycoprotein 1 IgA antibodies. We did a mixing study in this context, and we found a high patient and a low patient sample, and then mixed them in varying ratios. And here's what I'll show you here. Again, I'm only showing you data on one sample here. We took a high sample with a reactivity of about 130 units. We had a low sample with a reactivity of 22 units, and then we combined them in different ratios. measured the measured the reactivity that we observed, and then compared that to what the expected reactivity would be based on the ratio between the high neat and the low neat sample. And again, to try to be more simplified in the data I'm showing you, I didn't calculate any percent recoveries or percent differences here. However, I think you can appreciate the fact that what we're seeing on these measured values is very close to what we would see expected based on the theoretical mixing of this high sample and the low sample. And in fact, all of our percent differences here between these two values were within plus minus 5%. So certainly within what we would have expected. Another set of data I'd like to show you is proteinase 3 and anti-myeloperoxidase IgG antibodies. And on this particular data set, I'm showing you some dilution studies. And then I'm going to show you data for both proteinase 3 and myeloperoxidase on a couple of different samples for each. And I want to show you the differences that we saw here. So let's look at PR3 to start with. So for PR3, we started with a neat sample and then did serial dilutions in a 1 to 4, a 1 to 10, and 1 to 100. And then we calculated the reactivity based on that dilution to compare to the neat. And you can see for PR3 that for this first sample, when we did the 1 to 4 and the 1 to 10, that we got very close to the value we would have expected based on the neat sample, taking into account these dilutions. When we got to the 1 to 100, the signal was below the AMR, so we couldn't calculate it. We saw the same thing with the second sample where we started with a greater than 8 value. At a 1 to 4, it was still a greater than, which would have calculated at a greater than 32. However, when we got to the 1 to 10 and 1 to 100, we calculated very similar numeric values in our unit reactivities. In comparison, when we looked at the myeloperoxidase, we had one sample here where we started at a 6.3 at the neat, but you can see as we did the 1 to 4, 1 to 10, and 1 to 100, we had dramatically increasing reactivities that we were detecting when we took into account the dilution. This was not true for every myeloperoxidase antibody that we saw, because here we have a sample with a greater than 8 value on the neat sample. When we did the 1 to 4, 1 to 10, and 1 to 100, we saw very consistent results across those dilutions. So our take home from this is that the dilution responses may actually differ between patients or may actually differ between the antigen specificities. So what can I say about reportable range and how you verify that for autoantibody testing? Well, first of all, I think it's fair to say that when you do dilution or mixing studies, you should demonstrate a decrease in reactivity, although oftentimes it's not linear. Sometimes it is, as we saw with the beta-2 glycoprotein IgA testing with the mixing studies, that we had very, very good comparison between the measured and expected, but it's not always the case. What choice you use for the sample dilution, what matrix you use, is often it's often difficult to know this. Sometimes we use a negative serum, sometimes we use diluent, sometimes we use both. It really, I think, depends on the assay that you're looking at, the method, whether you have some negative serum that's available that might actually be more appropriate. But I think it sometimes worthwhile is that you may have to try both. Just as with precision, sometimes you are limited on the availability of the high sample, and so assessing linearity over the entire analytical measuring range may be difficult. There are recommendations that when you're assessing for linearity, that the sample that you use should be within a certain percentage. I think about 15% of the upper limit of quantitation. But again, you may not always have access to a sample like that. And so you may be limited to assessing for linearity just based on whatever samples that you have at your disposal. But I think lastly, I would say that sometimes you just need several approaches to really assess the analytical measuring range. You may need to do mixing studies. You may need to also do dilution studies. And I think it's often, I think, very important that you correlate this with your precision data, because again, the analytical measuring range really does tie to this idea that this is the range over which you are able to verify certain criteria of an analytical method, and that would be specifically precision. So correlating what you see on your linearity experiments with what you see with precision for the autoantibody testing, I think can provide valuable information when you're establishing the analytical measuring range for your assay. Now let's move into reference range. So reference range or reference interval, another terminology, is the range of analyte concentrations that you would observe in healthy individuals. So generally speaking, we would measure the analyte concentration in a cohort of healthy or non-disease control individuals, and then establish a central or one-sided 95th percentile. So the experimental approach, if you're verifying a reference range that might have been established by a manufacturer, generally using healthy controls, 20 individuals per category. And I put that per category in there because if you want to also establish a range in adults and pediatrics, you would need 20 in each of those categories. If you think there would be differences between males and females, 20 per category would be important, would be needed as well. If you're talking about establishing a reference range, usually 120 is considered a robust number for establishing a reference range. And again, that would be per category. So adults versus pediatrics, males versus females, etc. So let me show you, let me just come back to this. So this is something that we do very frequently when we are looking at autoantibody testing. And I'll just show you some data that we've generated from some reference range verifications in our laboratory for a couple of different autoantibody assays. The first one we'll start with is anti-CCP. So when we did our CCP IgG verification, we started with 95 healthy controls. We looked at the reference range that was suggested by the manufacturer, which is greater than or equal to 20 units as being positive. And when we did our reference range verification and we measured our healthy controls, we found that basically all of our tests, all of our results were less than 15.6, in other words, below the analytical measuring range for the assay. So technically, we verified this reference range because all of our healthy controls were less than fell within the negative range for this particular method. Our data for TTG, we took a similar approach, although because, again, we were verifying a manufacturer's range, we started with 20 adult healthy controls. The reference range for this particular assay was 4 to 10 units per milliliter being weak positive, and then greater than 10 being positive. When we measured the 20 healthy controls that we had a reference range, our reference range verification showed that 19 of our samples were undetectable or falling below the analytical measuring range at less than 1.2 units per milliliter. We had one sample that gave us a numeric value, but that was still less than the cutoff for weak positive. So again, all 20 of the samples that we measured have been classified as negative according to the manufacturer's range. Lastly, I want to show you data from antiphosphatidylserine prothrombin IgG testing. So in this case, we had a healthy control cohort of 120 individuals, and we had a reference range from the manufacturer of greater than 30 units being positive. When we conducted our reference range verification, we had a small number of samples were showing as undetectable or less than 9.4 below the AMR for the assay. The majority of the samples that we had were giving a numeric value. However, they would have still been classified as negative or being less than 30 under the category for the manufacturer had set for negative. However, we did find that we had a significant number, 13% of our samples actually were above the cutoff of 30 that was established by the manufacturer. So in this case, because we had our full 120 samples, we actually established a new reference range for when we implemented our assay. We kept the 30 cutoff as being negative. We established a borderline range, and this was based on the reactivity or the unit values that we were seeing in the cohort of healthy controls where the results were showing above 30. So we established from 30.1 to 40 as a borderline range, and then anything at 40.1 and above would then be considered positive. So What else can we say about reference range? When we're establishing or verifying a reference range for autoantibody testing, I think there is value in demonstrating that individuals without disease of interest are negative for the autoantibody reactivity. However, I think that what we have to keep in mind is that this does tend to maximize the sensitivity of the testing and may actually give us a somewhat lower specificity because the healthy controls that we see often are below the analytical measuring range of the assay. That said, as the example I showed you with phosphatidylserine prothrombin, I think there still is value in doing this because sometimes we do find methods where our healthy controls actually do tell us that the cutoff that has been established may not be completely appropriate for our testing population. There are other ways to do this, and this often can be very challenging, however, but we can move to the direction of establishing a cutoff which allows us to distinguish a negative from positive result. This usually requires that we have not only a healthy control population, but also a population of individuals with a disease of interest. This allows us to identify a cutoff or a point where we can have optimal distinction between healthy and the diseased population. In other words, we're establishing a cutoff based on both healthy and diseased population rather than the healthy population alone. And I think this really allows us to evaluate both sensitivity and specificity of the testing, but this does have a lot of challenges to it because we may not always have access to a population of individuals who have the disease of interest. Lastly, now I'd like to move into accuracy assessment and how we might assess for accuracy for autoantibody testing. So again, let's think about what do we mean by accuracy. Accuracy can be defined as the closeness of agreement between a measured value and the true concentration of the analyte. Well, based on that definition, I guess I would ask, can we really assess accuracy for autoantibody testing? And I think I would have to say no, because we really don't know the true concentration of the analyte or the autoantibody that we're looking at. Because we don't have certified reference materials, we don't really have traceability to a gold standard or certified reference methodology. So if we can't assess trueness, if we can't assess the true concentration of our analyte, how do we evaluate accuracy for autoantibody assays? And in large part, I would say we're really not assessing for accuracy when we do these experiments. However, there are some things that we can do that will give us, I think, a good handle or at least some understanding of the performance of our autoantibody testing. The classic method that people use is to do a method comparison, which can be useful, and we'll talk a little bit about that in a moment. We may be able to correlate one particular autoantibody with a related autoantibody, or we may be able to correlate with clinical diagnosis. And again, I'd like to show you some example data from accuracy assessments that we've conducted in my laboratory for verification of autoantibody testing and kind of illustrate some of these different points. So the first one I'll talk about is the anti-CCP IgG testing. This is a marker for rheumatoid arthritis. And when we began doing this testing, we started off by just doing a simple method comparison between two enzyme immunoassay methods. We collected a sample cohort of 275 patients, individuals, I should say. We had a total of 85 positive specimens based on the predicate enzyme immunoassay that we were comparing to, and 190 negative samples. And this is the correlation that we saw across those 275 samples. So our results showed that overall we had a concordance of 94.9% between these two different enzyme immunoassays, which I think is very good from a qualitative concordance perspective. But we did have these 14 discrepant results, and they're shown here. We had 11 which were positive on assay number 1 and negative on our predicate, and 3 samples which were positive on the predicate and negative on our the assay we were evaluating. And so we began to investigate what the nature of these discrepancies would be. Now, I'm going to show you 2 different examples of these discrepancies and start to think about how we might, again, investigate these discrepancies. So here are 2 different examples. Sample number 1, and I should say too that the cutoff for assay number 1 is less than 5, so anything less than 5 is considered negative. For assay number 2, a cutoff of less than 20, so anything below 20 is considered negative. So this first sample we see, on the first assay is giving a less than value, so it's considered to be negative. On assay number 2, it's giving a result that's I would say a borderline positive, just above the cutoff. I would say this is a discrepancy that I would classify as kind of being close to the cutoff. Is the reason you're seeing the discrepancy. I think sometimes these types of discrepancies may be reflecting differences in the calibration or how the cutoff was established between the two methods. And I think what's relevant to point out here is that if you adjust the cutoff of one or the other assays, you can start to improve the correlation between the two methods. Now, that's not to say that you should adjust the cutoff. It's just to say that if you adjust the cutoff, you might start to improve your correlation. Now, that is different from the sample number 2 that I'm showing you here. On sample number 2, we get a negative or undetectable result on the first method, but a significantly positive result on the second method. And these are what I would say are more substantial or significant discrepancies. Now, we can talk about why we see these discrepancies, and we'll get into that in a little bit in just a minute. Sometimes I think these discrepancies are reflecting differences in the antigen between the methods, or at least maybe it's more accurate to say it's how the autoantibody is recognizing the antigen between the methods, maybe explaining some of this discrepancy. Also relevant to point out that if you try to adjust the cutoff, it's probably not going to help you improve the correlation because you have such a significant discrepancy between the two methods. But you're left asking yourself, when you have discrepancies like this, which is the quote correct result? Are you seeing this as a true positive and this is a false negative, or vice versa? Is the sample truly negative and what you're seeing here is a false positive? Now, I think, though, we should probably stop in our discussion of how we assess for accuracy and really think about what do we mean by a false positive or true positive when we think about autoantibody testing in particular. So a false positive result generally is a test result which inaccurately identifies the presence of a condition or a disease. But with autoantibody testing, maybe we need to think about this a little bit differently, in the sense that I think you can have an analytical false positive, which is different from a clinical false positive. So what do I mean by these two? How am I distinguishing between these two? I would define an analytical false positive, meaning that you're detecting reactivity in your assay, even though the patient does not have an antigen-specific autoantibody that is recognizing that specific antigen. So that to me is an analytical false positive, in comparison to a clinical false positive, which means that the patient actually has an autoantibody against that antigen that you are detecting. It's just that they don't have clinically apparent disease at this point in time. What could cause an analytical false positive? It's difficult to say. Maybe there's some nonspecific binding. Maybe there's reactivity to a contaminating antigen that's present within the assay. But basically, there's something that is giving you reactivity that you are detecting, even though the patient doesn't have the autoantibody of interest. Whereas a clinical false positive, the patient has that autoantibody. This could be results from an autoimmune process that's happening. They may be early on in their disease process, but it doesn't take away from the fact that they actually have that autoantibody. And so from an analytical perspective, it's not a false positive, right? They actually have the autoantibody. They just don't have clinically apparent disease that goes along with it at this point in time. So when we come back to our CCP comparison here, then how do we start to distinguish and how do we start to understand the nature of these discrepancies? Are we looking at analytical false positives? Do we have clinical false positives? And when we did this testing and when we did this method comparison, we actually did also have diagnostic information available from these individuals. We ended up, when we did chart review on these patients, we had 98 individuals who had a clinical diagnosis of rheumatoid arthritis. We had 77 individuals who had another connective tissue disease that was diagnosed, and we had 100 healthy controls that we included. So when we use that information to calculate sensitivity and specificity clinically, we find that that we had almost identical sensitivity between the two methods that we were looking at, but the specificity of assay number 1 was slightly reduced in comparison to assay number 2, leading us to believe, or what we were able to, when we looked at the clinical diagnosis, is that at least some of these positive results that were generated on assay number 1, the patient did not have a diagnosis of rheumatoid arthritis. Now, does that necessarily prove that these are false positive results, or at least analytical false positives? I would say no, because the patient, these individuals still may have the autoantibody. It's just that they don't have clinically apparent rheumatoid arthritis. They haven't been diagnosed at this particular point yet. So even when you have clinical information, I think distinguishing between an analytical false positive or a clinical false positive can still be challenging. However, knowing how the sensitivity specificity compares across these two different methods in conjunction with our method comparison, I think provides a lot of useful information on the performance of these two different assays. Now I'm going to show you a different example where we took a different approach for demonstration of accuracy. And this is for the anti-phosphatidylserine prothrombin IgG testing. Now this is a non-criteria autoantibody that has been associated with antiphospholipid syndrome. And in particular, literature reports had suggested that the presence of an antiphosphatidylserine prothrombin IgG correlated with the presence of a functional lupus anticoagulant. So we collected paired serum samples from samples that had been tested for lupus anticoagulant. We ended up with a total of 69 samples. There were 13 samples where the lupus anticoagulant testing was positive, and the remaining 56 were negative. We then measured the phosphatidylserine prothrombin IgG in the paired serum, and then looked at qualitative concordance with the functional lupus anticoagulant testing. And this is what we observed. Again, we had an overall concordance of about 81.5%, which are shown here with these samples. However, we had a fair number of discrepant samples. There were 6 samples, and they were discrepant in both directions, meaning that we had 6 samples which were positive or showed the presence of a functional lupus anticoagulant where the PSPT was negative. On the other hand, we had several samples which were positive for phosphatidylserine prothrombin, which were negative for a functional lupus anticoagulant. Now, I will say that we did try to investigate these discrepancies, but it became very difficult. Number one, it's difficult because the lupus anticoagulant testing, there are a lot of subtleties to that. And we were relying on the coagulation specialists to help us make these interpretations on the lupus anticoagulant, whether overall they thought there was enough evidence to support the presence of this functional testing. But in some cases, the abnormalities that they were seeing on the functional lupus anticoagulant were fairly minimal, and that could explain some of these discrepancies possibly. On the other hand, some of these samples where the PSPT IgG was positive, but the lupus anticoagulant was negative, this may actually reflect the presence of some kind of inhibitor such as heparin, and that may explain why we see some of these discrepancies as well. But the point here is that, again, we were trying to assess accuracy of our IgG phosphatidylserine prothrombin by comparing it to a related test. And sometimes this, can be useful information, but it also can be sometimes difficult to understand when you see discrepancies between the results. The next one I'll show you, the last one I'll show you for assessment of accuracy, we looked at for our verification of anti-F-actin IgG testing. So most smooth muscle antibodies, which are associated with autoimmune hepatitis, show specificity for F-actin as the antigen. So in order to assess accuracy of our F-actin assay, we decided to collect samples which had been ordered for smooth muscle antibody testing, which is done by indirect immunofluorescence. We then took all of the samples and tested for the anti-F-actin IgG. Here's again our description of the sample cohort. We had a total of 173 samples. Sorry, I have that backwards. That should have been 124 were negative for smooth muscle, and we had 49 that were positive for smooth muscle. And here is our correlation. Again, we saw here are the samples where we're seeing qualitative agreement. That gives us an overall concordance of 79.8%. However, we did have, again, a significant number of discrepant results, both directions, where some samples were positive on the smooth muscle antibody test, which were showing us negative on our F-actin assay. And several samples, almost an equal number, were positive on the F-actin, yet showing no reactivity on our smooth muscle antibody testing. So how do we investigate these discrepancies? Well, we came back to the same approach that we took with our, when we had the CCP testing, which was to look at the diagnostic performance. Again, we were lucky that we were able to get clinical information on all of the samples that we had assessed, and we looked at sensitivity and specificity of the F-actin IgG at various cutoffs, the manufacturer's cutoffs, as well as other cutoffs as we were assessing what if we had a more optimal cutoff to distinguish positive versus negative. And then we looked at the smooth muscle antibody using different cutoffs, again, for the titer that we detect on the indirect immunofluorescence testing. And what we observed here is that, generally speaking, the F-actin IgG testing seemed to give us a better combination of sensitivity and specificity in comparison to the SMA testing. And the reason I say that is because if we, for example, try to normalize for specificity on the smooth muscle testing, to get a specificity of 98.7%, we needed to use a cutoff of 1 to 160. However, that gave us pretty low sensitivity compared to the F-actin IgG testing. At a similar specificity of about 97%, that cutoff gave us a much increased sensitivity in comparison to the SMA. So we were fairly confident that the F-actin testing, despite those discrepancies with the smooth muscle testing, was giving us pretty reasonable sensitivity and specificity from a clinical diagnostic perspective. Again, that caveat that I would mention here is that we had a limited number of confirmed autoimmune hepatitis diagnoses, only 14 in our entire cohort. Now, again, does that mean that some of those patients who maybe had a positive smooth muscle and a negative F-actin didn't actually have autoimmune hepatitis? It's possible. And I think, a further, a more robust evaluation for those clinical diagnoses would have been necessary to evaluate that. But it's just, again, trying to show how you might combine your analytical comparison between methods or between related autoantibodies and including clinical information. So just to summarize a little bit on points to consider in terms of accuracy assessment, we know that method comparisons have limitations. It's not really a true measure of accuracy, and you will see discrepant results, and they can be very difficult to troubleshoot and to really understand. Are you looking at false positives? Are you looking at false negatives? And how do you distinguish between the two? With all that said, though, I actually think that method comparisons still provide a lot of important information related to how your test is performing, because knowing how your test correlates with other methods or other assays will be very important in guiding clinicians and helping them understand results that they might see on one method versus another. What can we say about clinical sensitivity and specificity? I think while, again, this is important to do, it can be very challenging, especially when you have access to limited samples, especially when you have a diagnosis that's relatively rare. This can be very time consuming and very labor intensive to do chart review, to collect samples from patients with known diagnoses. Very challenging. But similar to method comparisons, if it's something you're able to do, I think it does provide a lot of valuable information regarding how this method or how your method's going to perform from a clinical diagnostic perspective. So that's the end of my presentation. I'll just summarize a few points here. We know autoantibodies are very heterogeneous, and that can lead to a lot of challenges for the clinical laboratory. We know there are regulatory requirements that we have to meet for assay verification, but how we approach these for autoantibody serology tests is not always clear. And I'll leave you with this last point. And this is something that I like to do when I'm designing a verification designing verification experiments for autoantibody testing. I asked myself two questions. First, the first question is, how do I fulfill the regulatory requirements? This is something that we need to be able to do. But beyond that, I want to ask the question, really, what do I need to know about this assay performance in order to make sure that I have both an analytically and clinically robust assay? And oftentimes we need to go beyond those regulatory requirements in order to satisfy ourselves about assay performance. But if you ask yourself that question, I think it will help you design really, really solid verification experiments that will give you confidence in your autoantibody serology testing. I'm just going to end by acknowledging the antibody immunology laboratory at the Mayo Clinic. I showed you a lot of data from verification experiments, and I don't have time to mention all of the individuals who were involved in the collection of that data and assisting with those assay verifications. But I just want to highlight that we have an amazing group of laboratory assistants, clinical lab technologists, technical specialists, quality specialists, development technologists, as well as our laboratory supervisor and assistant supervisor, who provide an amazing support and really are experts on these verifications and really do a great job in conducting these experiments and giving us the confidence in our assays as we do these verifications. And I also wanted to highlight my co-directors in the laboratories, Dr. Maria Wilrich, David Murray, and Ann Tebow are just phenomenal resources for me and always available to bounce ideas off of, which I really do appreciate. And I'd also like to thank you for attending this webinar. I hope you learned something about autoantibody serology, test verification, and I am certainly happy to answer questions either now or after the webinar.
Thank you so much
for your time.
And thank you, Dr. Snyder, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So let's get started. It looks like we have some great questions coming in from our audience members already. Our first question, Dr. Snyder, how might a lab troubleshoot method discrepancies that are identified during verification?
This is a good question, and I think one that anybody who does autoantibody testing will encounter at some point when they're doing verifications. And I would say there's just not a really easy answer. It so much depends on the specific autoantibody that you're verifying or doing a verification for. So probably the first thing that I would do when I'm troubleshooting discrepancies is to bring in a third method, even a fourth method, and really get a more global view of the nature of discrepancies. Is it one method that's an outlier? Is it just discrepancies across multiple methods? And not even just bringing different assays in, but bringing in different methods. If you have one that you did by an enzyme immunoassay, do you have access to a method that maybe is a chemiluminescent assay or a fluorescent immunoassay? Having different methods like that can also help to understand. When you have a method discrepancy too, I think it's really important that you have a very clear understanding of the different methods that you're looking at. What are the antigens that are used in those methods? How are they different from one another? Anything that you can learn about that type of aspect of the testing, I think will also help you troubleshoot. And the last thing, that you might want to do is think about related autoantibodies that might correlate with the ones that you're looking at. That can also help you understand, when you have discrepancies between methods in terms of, is one method correlating with a related autoantibody versus a different one. But I'll tell you something, I really think troubleshooting method discrepancies, it takes a bit of creativity. And sometimes you really have to think about what the best approach is, given the nature of the discrepancy that you're looking at, as well as the antigen specificity and the different methods that you're assessing.
Thank you so much, Dr. Snyder. Now, our second question coming in, what are some unique challenges associated with verification of indirect immunofluorescent assays?
This, I wish I had time to talk about this in my presentation, because it was something that I had really wanted to do, because I think there are a lot of challenges for indirect immunofluorescence assays. One, even just coming down to precision, there's a lot of, people might not agree in terms of what is considered appropriate precision for an indirect immunofluorescence assay. Is it plus minus 1 titer? Is it plus minus 2 titers? What do you think is appropriate in your laboratory, given the diagnostic application of that testing? What do you think, or what would you be most comfortable with? So that's one aspect of it. The one that I think about most frequently, and maybe this is largely in the context of ANA testing, but it can be applicable to other immunofluorescence assays as well, is that when you're reporting a pattern for a given assay, like we do for ANA testing, where we could report many different patterns, what do you do during verification to assess accuracy, reproducibility of those various patterns? Do you need to do verification for all of those patterns? Should they all be included as part of your accuracy? This can be challenging because some patterns are quite rare and can be difficult to access samples. But I think that, I think we have to think about that when we're verifying it, when we're thinking about an indirect immunofluorescence assay, because we are reporting multiple patterns. And I think we need to have confidence during our verification that we're, again, accurately able to interpret those patterns. And then there's always the question of, when you're doing that pattern assessment, should you be also assessing for correlations with the expected antigen-specific autoantibody, if that testing is available? That could also be something that you would include during verification. So just kind of scratching the surface a little bit on really what I think are some of the things that we might need to think of additionally when we're doing an indirect immunofluorescence assay.
Thank you so much, Dr. Snyder. And I want to thank our audience for these great questions coming in. Our next question, other than the required regulatory components, what other parameters should be evaluated during verification?
So there are some that we also evaluate. It does depend on the method. So one that we do fairly regularly when we're talking about a plate-based assay, 96-well plate assay, is we do an experiment called plate drift. So this is, you can kind of think of it as a really expanded precision assessment, but you're looking at it more across the entire plate. And I'll explain to you why we have done that. We have found in the past, especially when you have these methods on automated pipetters, that if you don't have the program set appropriately, you may end up kind of having one end of the plate incubate longer. You may have washing that sits longer on one end of the plate versus another, just because of the timing of the pipetters. And what that ends up looking like then, if you do a plate drift experiment, and the way we set up a plate drift experiment is that you take a single sample and you run it the entire across the plate. Every well on that plate has that sample in it. And what you'll see is that on a good plate drift, right, you should have consistent measurement. Usually we say it has to be within our interassay precision. You should see that across the entire plate. But occasionally, when we have had, I would say, less than optimal program on our automated pipetter, we'll actually find a consistent drift across the plate. Maybe either the sample kind of increases across the plate, it might decrease across the plate, it might actually decrease from top to bottom down the plate. And that's just telling us, that we should be looking at our programming, because it might be that we don't have consistent timing across the plate. And so that's something that we do pretty regularly when we have a plate-based assay like that. Some other parameters that we usually evaluate is carryover, especially if we're not having disposable tips, because, again, we're looking at very significant differences between positive and negative samples. And some patients can have extraordinarily high titer antibodies. So we need to make sure that whenever we have a patient who has a positive, that's not then carrying over into another tube or well on our plate. And so we do assess for carryover as well. And again, you can be limited sometimes with sample availability, but it's something that I think is worth trying with at least the highest sample that you are able to get. There are some others that we can sometimes we'll do. Stability could be one. Sometimes we do specificity testing to make sure we're detecting the appropriate antigen specificity if there are kind of related antigens that we could be having crossover between. So those are just a couple of the things we'll think about whenever we're making verification plans for an assay above and beyond what our regulatory requirements would be.
Thank you so much, Dr. Snyder. We have time for a couple more questions. About the discrepancies of assays, the result was less than 2 neg and the other assay's result was 177. Did you perform serial dilution for positive results? And how did you learn about which assay give the better results?
Yeah, so that was one of the discrepancies. I think that was on the CCP testing. So we did not do serial dilutions. That 177 was still within the AMR of the assay, so we didn't feel that was necessary. So we did not do that. In terms of which assay gives the better result, again, this is a very challenging question to answer. That's why we brought in that clinical analysis looking at the diagnosis for the patient. If I remember correctly, that result, the one that I'm sharing there, actually was a patient who did have rheumatoid arthritis. So at least it was that the positive result was consistent with the diagnosis. But that's not always the case. And again, just because the patient doesn't have the diagnosis doesn't necessarily mean they don't really have that autoantibody. It's certainly possible that they do. So, but those are again some of the thoughts I think we have to have whenever we're looking at these types of discrepancies.
Thank you so much, Dr. Snyder. We have time for one more question. When performing a method comparison to validate an assay, for example, ELISA kit on different platform, what acceptance criteria do you use for qualitative results or quantitative results?
Yeah, and this is so I think this is a great question because you're right. We do look at our results usually qualitatively and quantitatively. Now, qualitatively is really the only thing we can do if we're looking at different ELISA kits, right? And I would say, for acceptance criteria for qualitative, we're usually in that range of 85 to 90% is what we'd like to see. Now, that does depend too, though, on whether the assay has like weak positive borderline ranges, in which case we may, to simplify that qualitative concordance, we may kind of lump them with the positive or lump them with the negative, because the more ranges you're trying to get qualitative agreement across, the more challenging it can be. And so, we have to be realistic about those ranges and, really think about what the clinical utility of those kind of borderline results are anyway. And that's why we will tend to kind of lump them with either a positive or negative to get more of just an overall qualitative agreement. For a quantitative, I think the person who wrote the question is correct. We're only looking quantitatively when you have the same ELISA kit on different platforms. And I would say that, generally speaking, we usually say that, if we're doing like a passing Bablock linear regression, we're looking for slopes in the between 0.8 to 1.2 range, so allowing for about a 20% difference globally across the methods, and in our squared correlation coefficient of at least 0.9 is kind of what our guidelines are, and I think that's reasonable looking across different platforms, again with the same kit, with the same assay.
Dr. Snyder, I want to thank you for your presentation today and your important research. Would you like to provide any closing remarks for our audience before we go?
Well, I would just like to, again, say thanks for joining me today. I do autoantibody assay testing. That's my primary responsibility. And so it was really fun to put this presentation together. I hope you learned a little bit, and hopefully we'll take this back to the work that you do in your laboratory. And again, thank you for joining us today.
Well, I want to thank you again, Dr. Snyder, for your time. And I'd also like to thank LabRoots and our sponsor, Biorad, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.